Throughout our course in biochemistry, what we've seen is that the structure of our biological molecules ultimately determines their biochemical function. So, in order to carry out the diverse functions that we need in our bodies, we need a diverse array of different biomolecules. Now, in the case of proteins, we accomplish this by having 20 different amino acids with very unique chemical properties. Right? So we had some that were more nonpolar, some that were more polar. We had different charges, positive or negative. And so this allowed us to create an incredible array of proteins with all kinds of different functions. Now, when we compare this to our sugars, when we initially looked at our sugar structure and reactivity, what we saw is that we only have a couple of different functional groups. We have a carbonyl, either an aldehyde or a ketone. And then, of course, we have lots of alcohol functional groups on our sugars, right? Generally, each carbon, aside from our carbonyl carbon, will have an alcohol functional group. So, of course, this did lead to some important reactions. So, for example, we could form our Hemiacetals and Hemiketals, which we use to cyclize our sugars. And then we can also form our full acetals and ketals, which we use to link our sugars together. And of course, this is all due to the nucleophilic and electrophilic natures of our alcohol and aldehyde functional groups. Based on this, it's easy to conclude that our sugars are not very diverse. However, even though we have a limited number of functional groups, there are a number of different ways that our carbohydrates will create additional diversity within our carbohydrate structures. One way is in the different mechanisms we have for attaching our sugars together. So even with just one monomer, we can create a wide range of different structures based on how we attach those sugars together, which we'll talk about that more when we get into our different kinds of polysaccharides or sugar polymers. But the other approach is to modify our carbonyl or alcohol functional groups in order to add on additional reactive groups. In today's lecture, we're going to look at some of the common modifications that are made to sugars in order to add additional reactive groups. So we're going to split these into different categories based on the kind of modification that is made. The first potential modification that we'll look at is amino sugar derivatives. So amino sugars replace at least one alcohol with an amine-derived group. Right? So you can see here in the top left, we have beta-D-glucosamine. So this is just the glucose, one of our simple common sugars, with a single amine replacing one of our alcohols. You'll notice all these structures here in blue are different varieties of glucose that have been modified. And in the pink, we have some of our other common hexoses that can be modified as well. So we can add on our same amine group to a galactose to make a galactosamine, or we can add it on to mannose to make our manosamine. In addition to adding just our NH2 amine group, we can also add amide groups. So here we have N-acetyl beta-D-glucosamine. So we have our amine there from our nitrogen, but then we've linked an acetyl group, which is our two carbons here with that carbonyl, to create an amide. Now, as we're adding on these amino groups, so of course, we're adding on additional hydrogen bond donors. So our amine group has additional hydrogen bonding capability with our two hydrogens that can act as hydrogen bond donors. We also have slightly different reactivity compared to our alcohols, such as, for example, the increased basicity of our amines. Our amino sugars tend to be very important in structural polymers. So again, as we look at our polysaccharides or sugar polymers, we'll see a lot of different amino sugars used there as well. Our next key modification is deoxygenating our sugars. So again, normally our sugars will always have an alcohol attached to every single carbon. If we remove one of these alcohols, so we're taking off an oxygen, we can create a variety of deoxy sugars. So here we have beta-L-fucose, where we've taken off the alcohol from that terminal carbon there. or we have ramenose as another possibility here. Again, just a slightly different sugar structure based on how our stereoisomers are arranged, which then results in a different sugar, but still a deoxy form here. So fucose is found in some different glycoproteins and glycolipids. So these are structures made from combinations of our carbohydrates with either proteins or lipids, respectively. However, the most common deoxy sugar is deoxyribose, which of course acts as the backbone of our DNA. So our deoxyribose will cyclize into our typical DNA sugar that you're probably familiar with from your biology classes. The key difference between our deoxy sugars and our standard sugars is that we've essentially reduced the reactivity by eliminating our alcohol. So when we compare deoxyribose with our ribose, our deoxyribose tends to be less reactive, which then means that it's actually more stable compared to our ordinary ribose sugars. So of course, this is very helpful in DNA, where we want to have a very stable genetic code that will last for a long time. Phosphorylated sugars tend to be most important as metabolic intermediates. Now, when we add on a phosphate group to our sugar, there's a couple different consequences of this. The first is that it often creates a higher energy molecule overall, right? So when we have this large negative charge from our phosphate group, it destabilizes our molecule. So this enables our molecule to react and then potentially produce ATP for our cells to use for energy. In addition to increasing the reactivity of our sugars, the phosphate group will also increase the solubility and the polarity of our sugar. Now, of course, glucose itself, with all of our alcohol functional groups, is relatively polar. However, because it's also a fairly small molecule, it is able to diffuse through the membrane to some extent. However, when we add on this highly charged phosphate group, we now are much too polar in order to move through a membrane. So our phosphate then essentially acts as an effective mechanism to lock our glucose in our cells. So it will come into our cells either through transporters or potentially some through passive diffusion through the membrane. It will then be phosphorylated, where it will stick within our cell and won't be able to move out without being actively transported out of our cells. If you think back to organic chemistry, you might recognize that our aldehyde and alcohol functional groups can be very easily converted into a number of other functional groups through oxidation reactions. So aldehydes, of course, can be oxidized to carboxylic acids. Alcohols can also be oxidized to carboxylic acids if they're primary alcohols. And so through oxidation, we can create a variety of other modified sugars. So the oxidation of our sugars will produce acidic sugars. So these will then tend to be negatively charged at physiological pH. Of course, our carboxylic acid can also then form esters. So we can end up forming cyclized sugars with esters instead of our acetyl or ketyl groups. The consequence of this oxidation is in some ways similar to our phosphorylation, right? So it increases the charge, which then makes our sugars more reactive. It also increases their solubility and increases the possibility for interacting with other molecules through ionic interactions. As you can see from our structures here, there are many different ways that we can oxidize our sugars, right? So there's a lot of different possible oxidation pathways. In general, our alcohols are harder to oxidize, right? It takes a stronger oxidizing agent to oxidize an alcohol. So our aldoses that have aldehydes that can be oxidized will tend to be most reactive in these oxidation reactions. So oxidation of glucose is actually a very important reaction. So if we take glucose and react it with oxygen, we will usually need an enzyme catalyst for this. we can produce our oxidized forms. This is our gluconolactone. So the lactone refers to our ester form of glucose. So once we oxidize it into our carboxylic acid, it's then esterifying to create our ring structure. Now, if we intentionally oxidize our glucose, we can then actually take the products of this reaction and use it to detect our glucose concentration. So this is actually one of the mechanisms that we can use to measure blood sugar concentration. Blood sugar, of course, is a very important measurement to determine overall health. For example, high blood sugar concentration can be an indication of diabetes. If we want to use this glucose oxidation reaction to track our glucose concentrations, we would use a process something like this. So we would take our glucose, we would react it. Again, we'd use an enzyme to help to catalyze this reaction in the presence of oxygen. We then take our hydrogen peroxide product, and we can react that with some sort of colored agent. So in this case, we're using this O-dianosidine, which is faintly orange, but then when it's oxidized, it reacts to a bright orange color. So we also need a peroxidase enzyme to help catalyze this second step. So we convert to our lactone and our hydrogen peroxide, and we use that hydrogen peroxide to create highly colored compounds that we can then measure colorimetrically. So in lab, for example, you would just put your glucose into a cuvette and you could measure it in your standard spectrophotometer. Of course, in most medical settings, you would have that measurement occurring within a standalone device that would be used to measure that glucose concentration directly. Now, this sort of colored approach isn't actually commonly used anymore. So most of our portable glucose sensors tend to use an electrochemical method. So it's still using the same basic approach of oxidizing our glucose and using that oxidation reaction to track our glucose concentration. But rather than producing a color, it's instead producing an electrochemical change. As you're oxidizing glucose, you're reducing something else, and you're tracking that reduction in order to measure your glucose concentration. It's important to note here that this sort of analysis of glucose is only possible because our glucose has this free anomeric carbon, right? So in this case, we don't actually have our aldehyde initially, but we can easily linearize our sugar to create the aldehyde form, which then can be oxidized to eventually form our lactone, right? So our sugar actually interconverts between the aldehyde and the hemiacetal form in order to allow this oxidation reaction to take place. If our hemiacetal of our anomeric carbon here reacts with another sugar to form our acetal, so we would link it together to an alcohol to another sugar molecule, represented by my simple hexagon here. So in this case, I can't easily linearize this to form my aldehyde. So this guy can't easily break apart to form my aldehyde, and it no longer can be oxidized then to produce my carboxylic acid or ester products. So we have to have that free anomeric carbon that's not linked together with another sugar in order to carry out this sort of detection approach. We refer to this kind of sugar as a reducing sugar. So reducing sugar is any sugar that has a free anomeric carbon that can then linearize and be oxidized in that reaction. Ketoses are still often considered reducing sugars. That's because those ketoses can often be converted fairly easily to aldoses, and then they would react in that oxidation reaction. So of course, the term reducing sugars comes from the fact that when our sugar is getting oxidized, it has to be reducing something else, right? So in the case of our colorimetric test, it was reducing oxygen to our hydrogen peroxide that was then used to produce a color change and detect the presence of our reducing sugar. So our reducing sugars can be detected in a wide variety of different ways in order to help us detect our sugars. So Felling's test, for example, uses copper. So copper gets reduced, and we use that change in order to detect our sugar. Tollin's test uses silver. So again, reducing our silver with our sugar, and then detecting that change in the oxidation state of our silver. Both our colorimetric tests and our modern electrochemical tests use this similar sort of reactivity in order to detect our glucose concentration. Now, our body itself can also use this additional reactivity of our reducing sugar to produce some interesting products. So in the case of glucose in our blood, that glucose can actually react using that aldehyde of our reducing sugar with nearby functional groups, including amine functional groups on our hemoglobin. So this can then proceed through several steps to form what we call glycated hemoglobin, where our sugar is now permanently linked onto our hemoglobin. So we actually have a covalent bond produced. I mean, we could, of course, reverse the reaction and eliminate that glucose, but it's not going to come off without some sort of reaction. So if we have a high blood sugar concentration, that will push this reaction forward and produce more of our glycated hemoglobin. So glycated hemoglobin is produced at high blood sugars. Now, once we've produced our glycated hemoglobin, this can actually continue to react, right? So our sugar can react in different ways, and we can expand this kind of sugar structure that's attached to our hemoglobin. So this leads to what we call advanced glycation end products, which can actually then end up cross-linking, and it will eventually lead to damage to a lot of our different organ systems. So this sort of process of hemoglobin glycation is actually one of the dangers of having very high blood sugar concentrations, right? So if that glucose is just sitting in your blood doing nothing, it's going to end up reacting in unintended ways and damaging our hemoglobin. Detecting hemoglobin glycation can be important for a number of different reasons. First, it would enable you to determine if you're in danger of producing those advanced glycation end products that would then eventually cause damage to your organs. And number two, it can act as a sensor for your blood sugar concentration. So once your hemoglobin gets glycated, this will, of course, modify the size or the molecular weight of your hemoglobin to some extent, but it will also change the charge of your hemoglobin, right? So remember, it's attaching to your amine groups, which your amine groups will normally be positively charged on your hemoglobin. And so when you attach your sugar on there, you're actually going to convert that into the neutral form. So you then have your NH2 linked to your glucose, which I'll abbreviate as GLU, and is now neutral. So we can easily detect glycation using electrophoresis, so separating based on the charge, and of course the size would affect it here as well. We could actually estimate the percentage of total hemoglobin based on this small peak of glycated hemoglobin compared to our normal hemoglobin. So this, again, will detect our levels of hemoglobin glycation and provide a warning if our glycation is too high. And since our glycation will directly relate to our blood sugar concentration, this can also indicate that overall blood concentration. This, of course, would be a more long-term measurement, so it would indicate kind of whether your blood sugar has tended to be high in the past in order to create those glycated hemoglobin, as compared to our direct reactive tests that only indicate what the glucose concentration is at this given point in time. So as we can see here, there are a lot of different ways that we can modify our sugars in order to create more complexity in the kinds of interactions and reactions that our sugars can have. Some of these can be very beneficial, right? So we can reduce the reactivity of our DNA, for example, or we can increase the reactivity of our metabolic sugars, Some of these can also be detrimental, such as our hemoglobin glycation, where our sugar reacts with an amine group and then modifies our hemoglobin and ends up damaging our body. But again, overall, these will increase the diversity of potential applications for our carbohydrate molecules.